In part 3, we had seen the role of proto-oncogenes in the genesis of cancer. We had seen that proto-oncogenes encode proteins which promote cell division, and a quantitative or qualitative abnormality in a proto-oncogene can transform a normal cell into a cancer cell. But an abnormality in a proto-oncogene alone is not sufficient for malignant transformation. Generally, an abnormality in some anti-oncogene is also necessary to transform a normal cell into a cancer cell. And in this video, we are going to look at the role of anti-oncogenes in the genesis of cancer. As we had seen earlier, the proto-oncogenes encode proteins which promote cell division. These proteins are growth factors, receptors for growth factors, and signal transducers for growth factors. So all these proteins are related to cell division. They promote cell division. But we also possess some genes having an opposite function. Their function is opposite to the function of the proto-oncogenes, and these genes are known as anti-oncogenes. They are also known as tumor suppressor genes. The anti-oncogenes encode proteins which inhibit cell division, and their function is to prevent cell division from becoming excessive. So proto-oncogenes promote cell division and anti-oncogenes inhibit cell division. As long as there is a balance between the actions of proto-oncogenes and anti-oncogenes, the rate of cell division, the rate of cell growth remains normal. But if this balance is disturbed, if the balance tilts towards unrestrained, unregulated cell division, the cell can transform into a cancer cell. And this can happen because of mutations in proto-oncogenes and mutations in anti-oncogenes. When the anti-oncogenes become abnormal because of mutations, they lose their inhibitory function. Their normal function is inhibitory, but due to a mutation, this inhibitory function is lost, and this tilts the balance towards unregulated, unrestrained cell division. So we have on the left side a tumor suppressor gene or an anti-oncogene. It encodes a protein which is a growth inhibiting protein. Its function is to inhibit cell division. And as long as this gene is normal and the protein encoded by the gene is normal, the cell division remains regulated, it remains normal. But when there is a mutation in a tumor suppressor gene or an anti-oncogene, an abnormal protein is synthesized. This protein is non-functional. It cannot inhibit cell division, and this results in unregulated cell division, unrestrained cell division, and the cell can transform into a malignant cell. If we compare the properties of proto-oncogenes and anti-oncogenes, proto-oncogenes are dominant genes, whereas anti-oncogenes are recessive genes. This means that in case of proto-oncogenes, a mutation in one allele of the gene is enough to change its function. But in case of anti-oncogenes, the function will be changed only when both the alleles become abnormal. They get mutated. Some important anti-oncogenes include the p53 gene, the RB1 gene, BRCA1 gene, BRCA2 gene, NF1 gene, DCC gene, P16 gene, and WT1 gene. We will first look at p53 gene. This gene is expressed, that means the protein encoded by this gene is synthesized in nearly every cell. The protein which is encoded by p53 gene is known as p53 protein because its molecular weight is 53,000. Now this protein, the p53 protein, plays a very important role in cell cycle just before the synthetic phase or the S phase. This is the phase in which DNA is replicated. So the cell cycle is a cyclic process. It can be divided into M phase, G1 phase, S phase, and G2 phase. In M phase, which is actually mitotic phase, the cell divides. It divides into two daughter cells. This phase is followed by G1 phase or gap 1 phase. And after gap 1 phase comes the synthetic phase or the S phase, in which the DNA of the cell is replicated. Two copies of the DNA are formed. The synthetic phase is followed by gap 2 phase or G2 phase. This is the phase in which the cell is preparing to divide, and this is followed by the mitotic phase or the M phase in which the cell divides. The normal function of p53 protein is to make sure that the DNA is undamaged, the DNA is normal before the cell divides. So if p53 protein detects a damage to DNA, the DNA is abnormal. This protein stops the cell cycle. It arrests the cell cycle in the G1 phase. So the cell cycle will not proceed from G1 phase to the next phase, which is synthetic phase. This arrest of cell cycle is also known as G1 arrest. The cell cycle is arrested in the G1 phase. The purpose of G1 arrest is to give more time to the DNA repair systems to repair the damage to DNA. So we have got a number of DNA repair systems, and G1 arrest gives more time to the DNA repair systems to repair the damaged DNA. If the DNA repair systems are able to repair the damage and the DNA becomes normal, the cell cycle is allowed to proceed to the next phase, which is S phase or the synthetic phase. So DNA will be replicated. But if the DNA repair systems are unable to repair the damage, the DNA remains damaged. Then the p53 protein pushes the cell into an alternate fate. And this fate is apoptosis, that is programmed cell death. So the cell undergoes apoptosis, it dies. And because of this apoptosis of the cell having damaged DNA, the damaged DNA is not passed on to the daughter cells. p53 gene is said to be the guardian of the genome. It maintains the integrity of the genome. It acts like a watchdog to ensure that the cells having damaged DNA are not allowed to divide, and the new cells which are formed, the daughter cells, have normal DNA. So as long as p53 is normal, the gene is normal, the protein is normal, only those cells which have normal undamaged DNA are allowed to divide. But mutations can occur in p53 gene, and these mutations are seen in a variety of cancers, different types of cancers. In fact, p53 gene is the most commonly mutated gene in human cancers. Mutations in p53 gene are found to be present in about half of the human cancers. And when the gene undergoes a mutation, the protein, the p53 protein becomes abnormal, it becomes non-functional. And this loss of function plays a very important role, a key role in transforming the normal cells into cancer cells. Sometimes mutations in p53 gene are inherited from the parents. That means a child is born with a mutated p53 gene, and when this happens, when mutations in p53 gene are present in the germ cells, it results in a disease known as Li-Fraumeni syndrome. In Li-Fraumeni syndrome, the p53 gene is abnormal right from birth, and this results in a variety of cancers in different organs developing at a very young age. Another important anti-oncogene or tumor suppressor gene is RB1. When this gene becomes abnormal due to a mutation, a specific malignancy occurs which is known as retinoblastoma. Retinoblastoma is a cancer of retinoblasts in the eye, and this is usually a childhood cancer. The cancer develops in very young age. The RB1 gene encodes a protein which is known as PRB. This protein can be phosphorylated, it can be dephosphorylated. So it can undergo reversible phosphorylation and dephosphorylation. The phosphorylated form of the protein is the inactive form, and dephosphorylated form is the active form of the protein. And the function of active PRB is to inhibit cell division. So when PRB is dephosphorylated, it inhibits cell division. When mutations occur in RB1 gene, the protein PRB becomes abnormal. It becomes non-functional. And when it becomes non-functional, it cannot exercise its inhibitory role. The normal role is inhibitory, but this inhibitory role is lost when PRB becomes abnormal due to a mutation, and then the retinoblasts are transformed into cancer cells. As we have seen, the anti-oncogenes are recessive genes. So the protein encoded by these genes will become abnormal only when mutations occur in both the alleles of the gene. So in case of RB1 gene also, the protein PRB becomes abnormal when mutations occur in both the alleles of the RB1 gene. But one mutation in one allele of the gene may be inherited from the parents. So now only one more mutation is required in the second allele for the protein to become abnormal. And this second mutation can occur soon after birth, and therefore retinoblastoma can occur in childhood. So a mutation in the second allele of the gene, the RB1 gene, can occur in early childhood, and moreover, this second mutation can occur in a number of cells, a number of retinoblasts. So all the retinoblasts have got one abnormal allele right from birth, But if a mutation occurs in the second allele after birth, a number of retinoblasts can become abnormal and multiple retinoblastomas can develop. Moreover, the brothers and sisters of the child may also have inherited one mutant allele. So they are also prone to retinoblastoma. So if a mutation occurs in the second allele in brothers and sisters, they will also develop retinoblastoma. Sometimes retinoblastoma can occur even when one has not inherited a mutant allele from parents. And this type of disease is known as sporadic retinoblastoma. So this sporadic retinoblastoma occurs in the absence of inherited mutations in the RB1 gene. But for RB1 gene to become abnormal, two mutations at the same locus are required for the protein to become abnormal for development of retinoblastoma. This is not common. Two mutations at the same locus are not common, and therefore the sporadic disease, sporadic retinoblastoma, is rare, and the tumor usually develops at a late age. So mutations in p53 gene can cause malignant transformation. Mutations in RB1 gene can cause malignant transformation. But the proteins encoded by these two genes have a bearing on the cell cycle also. As we have seen, the cell cycle can be divided into M phase, G1 phase, S phase, and G2 phase. And the progression of cell cycle from one phase to the next is regulated by some regulators. These regulators include some proteins and some enzymes. And these regulators of cell cycle are affected by the proteins encoded by p53 gene and RB1 gene. Normally, when mitogenic signals are not present, mitogenic signals are mainly the growth factors. When growth factors are not present, the cell cycle enters a relatively quiet phase, which is known as quiescent phase. So in this phase, the cell cycle almost comes to a standstill. But when a mitogenic signal in the form of a growth factor is present, the cell cycle becomes active again. So from G0 phase, it can enter G1 phase, and then it will progress to S phase, it will progress to G2 phase, and finally to M phase, and the cell will divide. The proteins which regulate the progression of cell cycle are known as cyclins. There are different cyclins like cyclin A, cyclin B, and so on. And these proteins, the cyclins, affect some enzymes. In fact, they activate some enzymes, and these enzymes are known as cyclin dependent kinases or CDKs. And there are different cyclin dependent kinases like CDK1, CDK2, and so on. So cyclin D is an activator of CDK4, and when CDK4 becomes active, the cell cycle progresses from G1 phase to the S phase. Cyclin E is an activator of CDK2. Cyclin A can also activate CDK2, and cyclin B can also activate CDK2. And when CDK2 becomes active, the cell cycle progresses from S phase to G2 phase, and from G2 phase to M phase. When the cell cycle progresses through the four phases, that is M phase, G1 phase, S phase, and G2 phase, there are two important checkpoints. The first is known as G1 to S checkpoint. The second is known as G2 to M checkpoint. At G1 to S checkpoint, the regulators check whether all the requirements for synthesis of DNA are complete. And the most important requirement is that the DNA should be undamaged. The DNA should be normal. So G1 to S checkpoint allows the cell cycle to progress from G1 phase to S phase only when the DNA is undamaged. The second checkpoint, that is G2 to M checkpoint, is present late in G2 phase, just before the mitotic phase. And at this checkpoint, the regulators check if the replication has been completed successfully or not. If the replication of DNA is complete, the cell cycle is allowed to progress into the M phase. If the replication is incomplete, the cell cycle is not allowed to progress further. Now, G1 phase is the phase during which the cell is responsive to mitogenic signals, mainly the growth factors. And during this G1 phase, late in the G1 phase, there is a point in time which is known as restriction point or R point. This restriction point, which is present late in the G1 phase, is a point of no return. That means if the cell cycle has reached the restriction point, the cell has to decide either to proceed to the S phase, that is synthesis of DNA, or to remain in G1 phase. For example, the DNA is damaged. More time is required to repair the DNA. So the cell will remain in the G1 phase. And the third phase is the cell can enter the Z0 phase. It becomes almost quiet as far as the progression of the cell cycle is concerned. So at restriction point, the cell has to decide whether to proceed to S phase or to remain in G1 phase or to enter G0 phase. P53 is a very important regulator of G1 to S checkpoint, the first checkpoint. P53 protein can detect any stress, and damage to DNA is also a type of stress. So if P53 protein finds that the DNA is undamaged, a protein known as MDM2 attaches to P53 protein, and this MDM2 protein promotes the breakdown or degradation of P53. P53 is completely broken down. But if the DNA is damaged, some protein kinases which are present in the cell phosphorylate P53 protein. This is MDM2 gene in the DNA. This gene is expressed and the protein encoded by this gene, which is MDM2 protein, is synthesized. Now at G1 to S checkpoint, the P53 protein screens the DNA for any sign of damage. So the DNA will be either found to be normal, undamaged, or it will be found to be damaged. If the DNA is not damaged, if it is normal, P53 protein is not phosphorylated. And now this MDM2 protein binds the P53 protein. So a complex made up of P53 and MDM2 proteins is formed. And once this complex is formed, this MDM2 protein promotes the degradation or breakdown of P53 protein. So P53 protein is broken down. When P53 is not available in the cell, it cannot arrest the cell cycle. So cell cycle will progress from G1 phase to S phase. But if the DNA is found to be damaged, abnormal, then some protein kinases which are present in the cell phosphorylate p53 protein. And when p53 protein is phosphorylated, MDM2 protein cannot bind to p53. So p53 is not degraded, it is not broken down. And when p53 is present in the cell, it will arrest the cell cycle in the G1 phase. So phosphorylated p53 cannot be bound by MDM2. p53 becomes stable, and now this stable p53 increases the expression of a gene which is CDKN1A gene. The protein encoded by this gene is P21CIP1 protein. This is different from the P21 protein which is encoded by RES gene. RES gene is a proto-oncogene, and the protein encoded by RES gene is known as P21-RES or small GTPase. The protein encoded by CDKN1A is known as P21CIP1, and this protein is an inhibitor of cyclin-dependent kinases. So when this CDKN1A gene is expressed, protein P21CIP1 is formed, and this protein inhibits cyclin-dependent kinases. So if p53 protein is not broken down, it promotes or induces the synthesis of P21CIP1 protein, and this protein inhibits CDK4 and 6. It also inhibits CDK2, and when these cyclin-dependent kinases are inhibited, the cell cycle cannot progress from G1 to S phase, so there is G1 arrest. So P21CIP1 protein inhibits the cyclin-dependent kinases, and this arrests the cell cycle in the G1 phase, and this G1 arrest gives more time to the DNA repair systems to repair the damage to DNA. If the damage cannot be repaired by the DNA repair systems, then the p53 protein pushes the cell into apoptosis, so the cell dies. But if there is a mutation in the p53 gene, the protein p53 becomes abnormal, it becomes inactive, it cannot arrest the cell cycle in the G1 phase, and then the damaged DNA persists, and this can cause malignant transformation. PRB, the protein encoded by RB1 gene, also has an important role in progression of the cell cycle from G1 phase to S phase. In fact, PRB is the regulator of transition of cell cycle past the restriction point or the R point. A progression of cell cycle past the restriction point can occur only when PRB is phosphorylated. In fact, when it is hyperphosphorylated. When the G1 phase begins, PRB is dephosphorylated. And as we have seen earlier, dephosphorylated PRB is the active form of PRB. This form of PRB binds a protein known as E2F, which is a transcription factor. And when PRB is bound to E2F, E2F cannot perform its function as a transcription factor. Normally, free E2F binds to DNA and it promotes the transcription of certain genes, which is essential for progression of the cell cycle past the restriction point. But when PRB is bound to E2F and E2F is not free, it cannot bind to DNA. It cannot cause transcription of the genes necessary for the progression of cell cycle past the restriction point, and then the cell cycle is arrested in G1 phase. So for G1 arrest, we require two conditions. Number one, P53 should be present so that it can induce P21 CIP1 protein, and this protein will inhibit the cyclin-dependent kinases. And the second requirement for G1 arrest is that PRB should be dephosphorylated and active so that it can bind E2F, and when PRB is bound to E2F, G1 arrest occurs. Concentration of cyclin D increases in early G1 phase, and cyclin D is the activator of CDK4 and CDK6. So these two kinases are activated by cyclin D in early G1 phase, and these two kinases cause partial phosphorylation of PRB. Late in G1 phase, concentration of cyclin E increases, and cyclin E is the activator of CDK2. When CDK2 becomes active, it causes further phosphorylation of PRB. So this phosphorylation of PRB initially by CDK4 and CDK6 and then by CDK2 results in hyperphosphorylation of PRB, and hyperphosphorylated PRB is inactive form of PRB. PRB becomes inactive. The phosphorylation of PRB by cyclin-dependent kinases occurs when the cell receives mitogenic stimuli, and these mitogenic stimuli are in the form of secretion of growth factors. So when a growth factor binds to its receptor on a target cell, PRB is phosphorylated, and phosphorylation of PRB is essential for progression of cell cycle into the S phase. So binding of growth factors to their receptors on a target cell create mitogenic stimuli, and these stimuli increase the concentration of cyclin D and cyclin E in the target cell. The concentration of cyclin D increases early in G1 phase. The concentration of cyclin E increases late in G1 phase. Now, these two cyclins activate CDK2, CDK4, and CDK6, and these cyclin-dependent kinases cause phosphorylation of PRB, and when PRB is phosphorylated, it dissociates from E2F, and E2F becomes free to perform its function of transcription factor. Free E2F binds to DNA and promotes the transcription of genes necessary for progression of cell cycle past the restriction point. Once the restriction point is crossed, the cell is bound to divide. It is destined to divide. In this way, inactivation of PRB by phosphorylation is necessary for progression of cell cycle and for cell division. In the absence of growth factors, PRB is dephosphorylated. It is bound to E2F, and E2F is not free. But when growth factors are bound to their receptors on a cell, mitogenic stimuli are created, and these stimuli result in activation of cyclin-dependent kinases, and these kinases phosphorylate PRB. So PRB is hyperphosphorylated, it becomes inactive, and inactive hyperphosphorylated PRB cannot bind to E2F, it dissociates from E2F, E2F becomes free, it binds to DNA, it promotes the transcription and translation of the genes which are required for progression of cell cycle from G1 phase to the S phase. But if RB1 gene is mutated, an abnormal PRB is synthesized, and this abnormal PRB is permanently inactive. And when PRB is permanently inactive, it cannot stop the cell from dividing. The cell will keep on dividing. And this excessive cell division can lead to malignant transformation. So inactivation of RB1 gene by mutation leads to the formation of an abnormal PRB which is permanently inactive, and this causes malignant transformation. So here we have an abnormal PRB, a mutant PRB which is permanently inactive. It cannot bind to E2F. Now E2F is free. It can bind to DNA and it can transcribe and translate the genes which is necessary for progression of cell cycle from G1 phase to S phase. BRCA1 is another anti-oncogene. The name is derived from breast cancer because abnormalities in this gene result in breast cancer. BRCA1 gene encodes a DNA repair protein. This gene is expressed mainly in the mammary tissue. And since it is an anti-oncogene, the gene becomes abnormal only when mutations occur in both the alleles of the gene. And when both the alleles are abnormal, a non-functional DNA repair protein is synthesized, and this can result in breast cancer. A mutation in one allele of the gene may be inherited from the parents. So the child is born with one mutant allele, and now if a mutation occurs in the second allele after birth, the gene will become abnormal. It has been shown that women who inherit one mutant allele from parents have 90% chance of developing breast cancer because only one mutation in the second allele is required for the gene to lose its function, which can result in malignant transformation. BRCA2 gene is another anti-oncogene very similar to BRCA1 gene. This gene also encodes a DNA repair protein and it is expressed mainly in mammary tissue. If mutations occur in both the alleles of the gene becomes abnormal, the protein encoded by the gene becomes non-functional, and this can result in breast cancer. Again, mutation in one allele can be inherited from the parents. And such persons who have got one mutant allele from birth are known as carriers. And these carriers of BRCA2 mutation have a very high risk of developing breast cancer. Another anti-oncogene is NF1. The full name is neurofibromin 1 gene. This gene encodes the GTPase activating protein or GAP. We have seen earlier that this GAP is required to switch on the GTPase activity of P21 or P21-RES, the protein encoded by the RES gene. When mutations occur in NF1 gene, a non-functional GAP is synthesized. Normally, when a growth factor binds to its receptor on the target cell, in the P21-RES protein, GDP is displaced by GTP. When GDP is attached to P21-RES protein, the protein is inactive. When GTP is bound to P21-RES protein, the protein becomes active and it sends growth signals, the cell divides. Normally, P21-RES also possesses GTPase activity, but a protein known as GAP, GTPase activating protein, is required to switch on the GTPase activity of P21, and this active GTPase would hydrolyze GTP into GDP. Active P21 will be converted into inactive P21 and the cell will stop dividing. But when The GAP is abnormal. When GAP is non-functional due to a mutation, the active P21 res cannot be converted into inactive P21 res. So P21 res becomes persistently active, permanently active, and this leads to excessive cell division. It leads to malignant transformation. Mutations in NF1 gene cause a specific cancer, which is neurofibroma, and neurofibromas occur at multiple sites when there is a mutation in the NF1 gene. DCC gene is an anti-oncogene present on chromosome 18. The portion of the chromosome 18 having this gene was found to be deleted in a number of patients having colon cancer. And therefore, this gene was named as deleted in colorectal carcinoma gene. So DCC is the acronym for deleted in colorectal carcinoma. The function of normal DCC gene is to prevent carcinoma of colon. Now, deletion or absence of DCC gene results in carcinoma of colon. P16 gene is an anti-oncogene that encodes P16 protein. This protein is an inhibitor of CDK4. Normally, CDK4 is activated by cyclin D and it can be inhibited by P16 protein. And when CDK4 is inhibited, the progression of cell cycle from G1 phase to S phase is arrested. The purpose of this arrest, as we have seen earlier, is to give more time to DNA repair systems to repair damaged DNA. So when a cell receives mitogenic signals, P16 is synthesized. P16 inhibits CDK4. This inhibition prevents phosphorylation of PRB. PRB is not phosphorylated, and when PRB is dephosphorylated, the cell cycle can progress from G1 phase to S phase and the cell will divide. But when there is a mutation in P16 gene, an abnormal P16 protein is synthesized. It is a non-functional protein which cannot inhibit CDK4. And when CDK4 cannot be inhibited, the cell cycle cannot be arrested. Damaged DNA will not be repaired, and this can result in malignant transformation. Mutations in P16 gene have been seen in malignant melanoma and in cancers of the pancreas. Another anti-oncogene is WT1 gene. The name is derived from Wilms tumor, which is a cancer of the kidneys, and it is a childhood cancer. This cancer develops in childhood. This gene WT1 encodes a transcription factor which binds to DNA and which promotes the transcription of certain genes. But when the WT1 gene is mutated, an abnormal WT1 protein is synthesized. And this abnormal protein, the mutant WT1 protein, has a decreased ability to bind to DNA and it cannot promote the transcription of the genes which it normally promotes. This results in unregulated, unrestrained cell division and ultimately malignant transformation in the kidneys, causing Wilms tumor. So this is how mutations in anti-oncogenes result in the synthesis of non-functional proteins, which ultimately results in malignant transformation.